Okay, let's talk about exosomes. In aesthetic medicine, that word is absolutely everywhere, promising some truly revolutionary results. But if you look just a little closer, you'll see two completely different worlds. You've got the plant-derived products, and then you have the autologous systems that use a patient's own biology. The marketing, well, it can sound confusingly similar, but the science, worlds apart. So today, let's break down the facts and bring some much-needed clarity to the clinic.
Here's our game plan. We're going to start with a basic question of identity. What are these things, really? Then we'll get into the biology, layer by layer. After that, we'll follow the data to see what the numbers are telling us, take a hard look at the clinical evidence, and finally, we'll put all the pieces together to help you make a clear clinical choice.
So let's just start right there, with the name itself. The term plant exosome is plastered everywhere, but is it even scientifically accurate? As it turns out, in this case, the terminology, well, it's everything.
Let's set our gold standard right here. According to the absolute latest 2023 guidelines from the International Society for Extracellular Vesicles, the definition couldn't be clearer. To call something an exosome, you have to show it came from the endosomal pathway inside a cell. That's the scientific benchmark, period.
And here is the absolute core of the issue. Our own cells, mammalian cells, they use this very specific pathway. They create vesicles that carry these little protein ID tags like CD9, CD63, and CD81. Plant cells? They don't have that same pathway. They don't make those markers. So by the official scientific standard, vesicles from plants, they just aren't exosomes.
And listen, this isn't just some academic squabble. This has real-world consequences. In South Korea, which is the home market for a lot of these products, regulators have actually banned the word exosome in cosmetic ads. Why? They decided it was misleading to consumers. That action, coming from the manufacturer's own backyard, is pretty telling.
Okay, so we've tackled the name. Now let's dive deep into the actual biology. We're going to break this down into three distinct layers: the container that's doing the delivering, the cargo that's inside it, and finally, the cost of the whole system. Think about this delivery system like a key and a lock. Our own extracellular vesicles are EVs. They have these specific proteins on the surface. These are the keys. They're designed to fit perfectly into the receptors on our cells, which are the locks. This gives you targeted, efficient delivery. Now, plant vesicles, they have plant keys. And right now, there is zero published evidence to show that a plant key can open a human cellular lock. The package might show up at the door, but there's no proof it can actually get inside. Now for layer 2. Let's look at the cargo. What's inside? You know, real tissue regeneration relies on very specific human signals. PDFT to bring in the fibroblasts, TGF-beta1 to kickstart collagen production, and VEGF to build new blood vessels. A plant, say a rose, it just doesn't have the genetic blueprint to make these human growth factors. Biologically speaking, it's impossible, which means the most critical regenerative signals, they're just not in the vial. Now, the big counterargument you'll hear is that the active cargo isn't growth factors, it's these bioactive polyphenols. And okay, fair enough, polyphenols have known benefits. But this brings up a huge paradox. These molecules are freely soluble. You can just apply them directly. So the question is, why would you pay a massive premium for an unproven, uncharacterized delivery truck to carry something that doesn't even need a ride? Yeah, let's just put this in simple economic terms. You can deliver incredibly potent polyphenols, like vitamin C, with a $15 serum that uses a well-understood topical pathway. Or you can spend $200, $300, even $400 on a vial that uses a delivery mechanism with no known uptake in human cells. The value proposition here gets really, really hard to justify. But what about plant mRNA, you might ask? Well, this idea that it can talk across species, so-called cross-kingdom communication, has been debated for over a decade. The original findings back in 2012 were immediately challenged, and they've never really been reproduced reliably. Fast forward to today, and the scientific consensus is shifting. If there is any effect, it's likely just a general immune response, not the specific, targeted gene-level work needed for true regeneration. After all this time, there's still no solid clinical proof. Okay, now for what I think might be the most revealing part of our entire investigation. For a minute, let's just forget about the type of biology. Let's just talk about pure quantity. This is all about the dose. These numbers on this chart tell an absolutely incredible story. A typical plant-based vial, you're getting maybe 5 to 10 billion particles. Now look at that middle bar. That's a single milliliter of your patient's own plasma, completely untreated, straight from their body. It already has anywhere from 140 billion to over 8 trillion particles. Now look at what happens when you concentrate that plasma with a system like Exosmart. You're reaching densities up to 30 trillion particles per ml. The difference in scale, it's just staggering. Let me just put a fine point on that, because it's wild. Before you do anything, before you even uncap a product, your patient's own blood in a single milliliter already contains 14 to 240 times more particles than what's in that entire expensive vial. Their own baseline biology is already way more concentrated.
So what happens over a full course of treatment? Well, 4 sessions with a plant product might get you a total of 20 to 40 billion particles. A single session with a concentrated autologous system can deliver around 195 times trillion. That's not a typo. This creates a dose gap of 5,000 to 10,000 times. And most importantly, it's a higher dose of the right stuff, the patient's own biology. So, with all that context, the questionable biology, the tiny dose, let's look at the clinical evidence. Is the data solid? And maybe just as important, where is this whole category heading? This number is pretty shocking. If you combine all the published studies for every single plant-based exosome product on the market, the total number of human subjects studied is just 246. For an entire therapeutic category that's making these huge claims, that is an unbelievably small base of evidence.
Okay, let's break down the best evidence they have. The most cited study, their only randomized controlled trial. First big problem, it was registered retroactively. That's a major red flag in clinical research. Second, the product was mixed with other active botanicals, which makes it totally impossible to say the effects came from the vesicles. And finally, the study didn't even provide data to prove what was actually in the vial.
This slide shows you exactly where this product category is headed. It started out as this premium, professional-only product. Pretty quickly, the same companies launched direct-to-consumer sprays. And today, you can find dozens of these so-called exosome products on Amazon for less than $30. The trajectory isn't towards rigorous clinical trials. It's towards the mass-market cosmetics aisle.
So let's pull all of this together. At the end of the day, as a physician, you have to make a choice, and that choice has to be based on the data. The biology should be what informs your decision.
Here it is, side by side, the two options. On one side, you have a cosmetic product. Its plant biology has an unknown uptake mechanism, a very low particle count, and 0 human growth factors. On the other side, you have a medical device. Its human biology has known receptor-mediated uptake, a massively higher particle count, and verified human growth factors.
And let's be really clear here. The demand for exosome therapy is real. The plant-based category did an amazing job of building a market and getting patients excited about the idea. They proved the demand is there. So the opportunity now is to actually deliver on what that market was promised in the first place: real, potent, human regenerative biology. So we're going to end with this question. A patient walks into your office, they're excited, they're hopeful, and they're asking for the cutting-edge exosome therapy they've heard so much about. They are trusting you to make the best choice for them. When you look at all the evidence, the naming, the container, the cargo, the dose, and the data, which vial are you going to reach for?
Enjoyed this video? Like, share, and subscribe for more evidence-based content.